Hi everyone, welcome to the channel Botany with Sadhna. In this video, we will discuss how anatomy of dicots are different from monocots. So first of all, we will discuss about anatomy of dicot root. In dicot root, the outermost layer is called epidermis, which are having which is having unicellular hairs, or we can say that root hairs or epidermal hairs. Up below that, few layers of cortex is present, made up of parenchymatous cells, and the innermost layer of cortex is the endodermis. Endodermis is different from the cortex in the shape of cells present. Endodermis is barrel-shaped cells of parenchymatous cells. After that, in endodermis, pericycle is present, which is few layered thick, and from here vascular bundles are present. Xylem and phloem are present. Xylem is in condition of exarch, means protoxylem is towards the periphery, whereas metaxylem is towards the center, and the phloem is present near to the xylem. As we have seen that in dicots, radial arrangement of vascular bundle is present, which means that xylem and phloem both are present on different radius. So here in dicot root, one of the characteristic feature is we are having only 2 to 4 patches of xylem, means we are having only 2 to 4 patches of xylem, and the pith is small in nature. And now, how it is different from monocot root? In monocot root, the other like epidermis, cortex, endodermis is present, is similar to the dicot, but the difference came here, and the pericycle is also present inside the endodermis. Here, the pith is larger in size as comparative to dicot root, and the xylem patches are present in 6 to 8 in numbers, whereas metaxylem is present towards the towards the center, and protoxylem is towards the periphery, and phloem is present near the xylem. But the main difference it is having 6 to 8 patches of xylem, whereas in dicot root we are having 2 to 4 patches of xylem. Next is how dicot stem is different from monocot stem. In dicot stem, the outermost, as we everyone know, outermost layer is epidermis, having trichomes, or we can say that epidermal hair-like growth. In stem, it is called as trichomes, whereas in roots they are called as root hairs. Trichome. After that, below epidermis, few layers of cortex is present, which is divided into 3 parts: hypodermis, then starch present in the between the layer, and the innermost layer is called endodermis. So cortex is divided into 3 parts. Hypodermis is made up of sclerenchymatous cells, whereas below hypodermis, starchy sheath is present, which are having reserve food material as starch, and the innermost layer is endodermis. After that, pericycle is present, which is parenchymatous in nature. Parenchymatous cells are present in pericycle, and after that, phloem is present. Phloem, and then a ring of cambium is present, which is a characteristic feature of stem, or we can say that dicot stem, that cambium is present between the vascular bundles, means phloem, xylem and phloem. After that, xylem is present in the condition of endarch, means protoxylem is towards the center and metaxylem is towards the periphery. So enlarged condition is seen and the pith is large, means, and as we see this is a small transverse section, and when we see the whole transverse section we see that the cambium ring is present, which is a characteristic feature of dicot stem. So here the ring is the characteristic feature of dicot stem, and cambium is present between xylem and phloem, and Dart's condition is seen in protoxylem and metaxylem. So this is, and one of the main important character is in dicot stem conjoined and open vascular type of bundles are present, which means that conjoined means xylem and phloem both are present on the same radius, and open means cambium is present between xylem and phloem. Now we will see how it is different from monocot stem. In monocot stem, epidermis, as we all know, epidermis is the outermost layer in every cell or in every organ. Epidermis is the outermost layer, then the hypodermis is present, which is sclerenchymatous is nature. Then vascular bundles are present in the form of scattered patches. Here cambium is absent and pith is also absent. Only ground tissue is present, where its ground tissue is present everywhere, and vascular bundles are present, are scattered here and there in the form of patches. So the main characteristics are of monocot stem and dicot stem is vascular bundles are present in the form of patches, and uh, they are scattered in the ground tissue. Whereas in dicot stem, it is present in the form in a proper arrangement with demarcating character in between them is cambium. So the dicot and monocot stem is the how, as we see here, the monocot stem and dicot stem are different. Next is anatomy of leaf. First of all, we will discuss about anatomy of dicot leaf. Dicot leaves are also called as dorsiventral leaf because number of stomata are different on the both the surface of leaf, and on lower surface we will call it as adaxial, whereas upper surface is called as abaxial. And lower surface in dicot leaf, lower surface are having stomata, whereas upper surface is having a few or very less stomata. The outermost layer is epidermis, here both on the abaxial surface and the adaxial surface. Next, below the epidermis is the mesophyll cell, which is in dicot leaf. It is differentiated into two types of cells. First is palisade mesophyll and then the spongy mesophyll. Palisade mesophyll is made up of parenchymatous cells, which is having no intercellular space and a proper arrangement of cells are seen. Whereas spongy mesophyll is having a large space between them, and air cavity is also present between the spongy mesophyll cells. And the vascular bundles are present in the inside the bundle sheath cell. Bundle sheath cell is a ring-like structure inside which vascular bundles or xylem and phloem are present. Phloem surrounds the xylem, means xylem is surrounded by phloem, and on the lower surface stomata are present, and we see that stomata is the here this is the stomata. These are guard cells. The cells surrounding the guard cells are subsidiary cells, and the cells surrounding the stomatal pore is the guard cells. So first of all, we have seen that the stomatal pore and the cells surrounding are guard cells, and the cells surrounding the guard cells are subsidiary cells, here, like this. And air cavity is also present so that proper exchange of gases can take place. So this is all about the anatomy of leaf. Now we will see how it is different from monocot leaf. In monocot leaf is also called as isobilateral leaf because on it is having both stomata is present on the both the surface lower and upper surface of leaf. Here stomata is present on the both the surface of leaf. The outermost layer is epidermis. Next, phloem is present on both the surfaces below the epidermis. Phloem is present and xylem are present on the in some scattered patches between the phloem. Or we can say that here also xylem is surrounded by phloem. And the one of the characteristic feature is also here mesophyll is not differentiated or not different is not characterized into different mesophyll cells, it having same kind of cells. So and here some cells at the upper epidermis, or we can say that upper layer of epidermis is modified into form of bulliform cells, and it is present in grasses where bulliform cells, whenever they absorb water or they are turgid in nature, the leaf surface is exposed. Leaf surface is exposed. Whereas whenever bulliform cells are flaccid or deficiencies of water or is present in them, then leaf shrinks. So here in this condition proper photosynthesis takes place, whereas in this condition transpiration is low. Here transpiration is also taking place in a full manner, whereas in flaccid condition transpiration and exchange of gases is also halt. So in this video we have seen that how dicot and anatomy of dicot and how anatomy of monocot is different from anatomy of dicots. I hope it is now clear to you all. Thank you for watching.